also go live here on broadcast. Now we are and it takes a little bit for people to stream in. So let's give that a minute until you see the counter count up. Oh, and now YouTube wants to will annoy me while playing back what I. Okay, great. Okay, let's give it another minute or so. It's funny, you can see like the count go up. And I think that's because everyone, before they're actually placed in here, need to like push a button that says that they want to share their audio or something. In any case, why don't we get started? Good afternoon, everyone. My name's Konrad Kording. I am almost brain dead because this day I started moderating on the Asia time zone, and then I moderated the European time zone. And okay, it wasn't actually so hard. Like I was really a panelist on the Asia time zone, and I was like only a co-moderator in Europe, and I'm also only a co-moderator now. And it's with great pleasure to introduce my co-moderator, who's Maryam Vaziri Pashkam. Maryam, why don't you introduce yourself?
Hello, everyone. I'm Maryam Vaziri Pashkam, and I work at NIMH. I'm a cognitive neuroscientist. I study visual cognition when it connects to action. I study how do we process visual information for action. So I spend a lot of my time like designing 3D printed objects and having people grasp them. And fun fact about me, when I want to get a break from science, I play a Persian instrument and sing Persian traditional music.
Wow, what kind of an instrument do you play?
There you go.
Wow. Awesome. Well, this is so cool. This is one of the things that I really love about Neomatch Academy, that like you really get to also experience the people that are behind everything. So, and we will be starting the Q&A very soon. But before that, I have to do what I've been instructed to do, which is remind everyone to do your reflections. We are currently in time zone 3. I want to applaud Saffron Pillbug. Well, I'm not quite sure what a pillbug is, but I should do the lookup of that just to honor that you guys all are doing your reflections. But you would do us just such a great favor if you could fill out the reflections. Primarily because, well, we're doing this for the first time. We need to learn how to do it better. And there's so many ways how we can do it better. So we will now spend another minute or two in awkward silence or useless chatter while everyone gets the chance to do their reflections. Also, please put your questions already in there. Like we have the Q&A button, which allows you to ask questions and vote on questions. Voting on questions is really important because otherwise you will be answering the wrong questions. Okay, let's give them another 30 seconds or so, no? So these are always the longest 30 seconds of my day when I have to do the reflection slide. Thanks everyone for giving us the reflections. It really makes a big difference. So now, after encouraging you to do your reflections, I think it's time to introduce the 3 awesome Q&A panelists that we have.
Okay, Manjari, why don't you start?
Hello, everyone. I'm Manjari Nayaran. I'm a postdoc at Stanford School of Medicine. My background comes from I started off in electrical engineering. I moved on to statistics, became very interested in the sort of challenges of studying complex systems and human neuroscience. And so now I work on statistical methodology and causal inference in the context of human brain imaging, particularly functional MRI, but also interested in DTI, brain stimulation, and fMRI, brain stimulation, and EEG, and so forth. Glad to be here today. Yeah.
Thank you. And Sofia?
Hi, I'm Sofia. I work at the University of Pittsburgh. I design algorithms for causal discovery. And the fun fact about me is that I have been baking my own bread before the 2020 pandemic, far long before the 2020 pandemic, many, many years.
Thank you,
Awesome.
Sofia. Bijan?
Hi, I'm Bijan Pesaran. I'm a professor at NYU, and I'm working my lab studies the primate brain, non-human primates and humans. I entered the field from physics. I was analyzing data. Data is a wonderful thing, and it's been 20 years or more since that time for me. And I often feel that I would prefer to be a graduate student now than 20 plus years ago, but we get where we are from where we come from. And what's wonderful about today is that we have so much more data.
Yes.
And so my lab is focused on studying the monkey brain in detail with large-scale technologies to acquire vast quantities of data from animals performing tasks which require flexibility and planning, decision-making. And the other thing about data is that you can process it, and if you make it into a real-time interface, then that data can speak to you directly and be interpreted on the fly. And so that real-time processing is also becoming more powerful than it used to be. In concert with the large volumes of data we're collecting now, it becomes complicated. And so I think this is really what's exciting, is when we build interfaces that acquire and interpret vast quantities of data in monkeys and humans. And so that's why we have human studies in our lab. And fun fact, I ride my bike most days. And if you come to Central Park, we will meet. I look forward to seeing you in Central Park.
Okay, thank you, Bijan. So let's dive into the questions, everyone. So I'm going to start with the first one, which is a question about the material in the course, and I think it's a good question. It's, can you give us examples of instrumental variables in neuroscience? And who should start this? Maybe Sofia?
I think I'm the least qualified in this panel to talk about causality in neuroscience. Because I really don't know of any good examples.
Feel free to pass it to someone else.
I think either Konrad or Bijan should talk about.
I think Manjari is technically doing very related things, no?
So, well, I have some ideas on this. One, any kind of source of, external manipulation to the system is a potential candidate for an instrumental variable. But the key thing is that the instrumental variable should be either highly correlated or causal to the cause of interest, but it should not have any consequence on potential confounders that you're worried about. So it needs to mimic the ideal experiment, even if it isn't one. And I think trying to find these is actually a very interesting and useful problem, and that's something, I spend a lot of time thinking about. So one that comes to mind is the fact that you can, for those who do human neuroimaging, especially in functional MRI, one of the challenges in functional MRI is that one can have non-bold fluctuations in the magnetic field that contribute signal that you observe, but they're not biological, and they might yield correlated activity between brain regions that are of non-biological origin. And so if you want to get at that kind of information with the ground truth, it turns out that there are MRI sequences that are much better at manipulating the bold content of the fMRI signal without having any effect on the non-bold content of the fMRI signal. And so one, it doesn't occur to everybody, but like the instrumentation itself can be a source of instruments to get at, some aspects of the signal that you're interested in that are currently confounded. The other example would be, transcranial magnetic stimulation or any kind of external perturbation to the brain as a potential candidate. But as we might get into, just doing brain stimulation alone might not be an instrumental variable because it very much depends on what your causal question is. And so we can't off the top of our head say that it is an instrumental variable, but it could be for the right question.
And if I can add something here, there aren't many applications of instrumental variables in neuroscience. Why not? Because neuroscience hasn't, in most areas, thought all that much and all that hard about causality, that bridge between the causal inference literature in statistics and neuroscience hasn't existed for such a long time. And in that sense, the number of people who have run instrumental variable studies and that think about the brains, that intersection isn't very large. And if I may add an example, if we say have optogenetics, and let's say we're in a dish, we stimulate a small number of neurons, maybe 2, maybe 3 or something, then we have confounding. Now, like the same light will affect all the neurons. The questions, are there ways of using instrumental variables to still discover causal interactions between neurons? And we believe that there is, and we have a preprint somewhere about that, with the idea that if we have, that arguably if the neuron is currently refractory or is just not refractory or is just outside of its refractory period, that's arguably a reasonable random variable. It only affects the neuron itself, and it doesn't affect the rest of the network. And so we believe that we can figure out causal interactions between neurons by focusing on instrumental variables. But this is not an established approach.
So,
Can I add to that?
Bijan, go ahead.
So I completely agree with what Konrad is saying, that the links between the different literatures need to be developed. And I think when I went over the lectures today, I was struck by how this is clearly pregnant for future work. I was also struck by how our recent work directly relates to this, and we never use the term. We have a paper online at Neuron. Xiaoyu Xiao is the first author, who's now working in Scopics as a postdoc in my lab, QIAO. And July something, it came out literally just recently. And it's causal network analysis is what we present. And the heart of the idea is to have an instrumental variable. The idea is to have a minimally perturbative manipulation. And what is minimal? Well, that's a very complicated thing. It should be big enough to have an effect that we can measure. But it should not be so big that that effect is confounding. And what we could do was then study the response of the brain to that minimal perturbation, because we had an effect that was big enough to measure, but not so big that it was always there. And that was how we defined it. But there are so many questions that come out of those kinds of experiments because we did electrical microstimulation. We can do that anywhere in the brain. And we did this on tens of thousands of sites, large scale, as I already said, about our interests. And then we recorded neurons and field potentials all over the brain, but the neurons were kind of sparse, and we didn't have neuropixels that we now have with our thousands of neurons, and we had only microelectrodes with our individual neurons. And so we couldn't really find, we found some examples, but we didn't find across the network single neuron responses to precise manipulations. That would be what you would want if you could find it. Instead, we had electrical stimulation, which has kinds of recruitment effects involving fibers, and the response of that stimulation being measured with a field potential, still quite focal, it turns out, but it's not the same as a cell.
Bijan, can you briefly tell us what recruitment effects are? I don't believe everyone will know the term.
When I say that, I'm referring to just the fact that you're bringing many responses at play when you stimulate electrically. There are lots of different parts of the cell that you bring in. I don't mean the more complex effects of trains of stimulation. We were actually doing single pulses, one 200 microsecond pulse, biphasic. 10 microamps, 20 microamps, really a small amount of current going in the brain, we would say, once. And then you could see the effect of that. There are other effects when you do trains, which we avoided because we didn't want recruitment effects, but where you have trains of stimulation that you deliver, and then you get you recruit inhibition is what happens when you activate the neural circuit. The first thing you do is you recruit inhibition after that point. The refractoriness plays into that, of course, the tendency of cells not to fire again. And so those effects we didn't want to have. So we avoided those recruitment effects due to pulse trains, even though if you're recording somewhere else in the brain, do you really expect to see the effect of a tiny pulse of activity in the orbitofrontal cortex? Can you expect to really measure this in the striatum or in the anterior cingulate or in the prefrontal cortex, the dorsal we weren't sure. And so we would have used trains to get the effect that we could then study as a less minimal but still measurable perturbative response. And we would have used it, but we didn't have to do this. This is the point. We brought it down as little as possible. And this was the key. It was minimal as much as we could make it. And so, yeah, we never talked about instrumental variables the whole time. But this is absolutely an instrumental variable because you can analyze the response in a regression. And then what we could do is ask what predicts this response, which is now a variable that we are measuring through the minimal perturbation. We don't know what the response is going to be every time. That's the point. And then we measure what predicts the response. And then we gave that a name. We call that a modulator. And we call this, we have a whole language for this, of course. It's a causal network analysis. It has causal network mechanisms.
And you don't call it stage 1 and stage 2 of instrumental variables?
No. I think that we might not, but maybe we should. I'm not sure.
Manjari, you were also going to say something about the question, no?
Yeah, I wanted to refine it to say that, I became very interested in this when I was trying to figure out what we can learn about transcranial magnetic stimulation. There's a huge hope in human neuroimaging that rather than just observe brain activity without doing anything like in resting state, if you do single pulses or a train of pulses to different regions in the brain and observe what happens, you can learn causal networks. Now, the challenging thing about this is that we're trying to study causation in networks, and networks are not isolated systems. And so even if you do an intervention to a node in a network, it has downstream consequences to other parts of the network, which means that you're going to have a hard time separating who was directly affected and who was indirectly affected. And so there's a whole literature on this and doing instrumental variables in networks. And one of the ways of getting around this is if you're willing to relax your causal question to saying, rather than trying to understand whether region A causes region B, can you say there are groups of regions? I know that when I stimulate region A, there's a whole cluster of regions that are likely directly affected. But hey, I know that there are these other regions that are at least, 2 hops away. Now I can treat my perturbation to a region as actually perturbation to a group of regions. And I can treat that as an instrumental variable for the manipulation of some other downstream regions. And so if you're willing to relax the resolution of your causal question, I might actually be able to tease apart causal relationships between large-scale communities in the brain, but not individual regions. And so recognizing, what is your instrument going to be an instrumental variable for can help, is also a different way of trying to find instrumental variables.
I want to ask another sort of clarification question about the course material that is a little further down. Emily Avery Jones asked, why do causal inferences methods fail in larger, denser network? I think if we answer this, then I think we can go back and answer a couple of the other questions that are before this. Who wants to take this?
It's like totally a Sofia question.
Exactly.
Sorry to put you on the spot.
Well, I mean, there are two reasons that you could think about that. First of all, the larger the system, it's not actually just a function of how large the system is, but how large the thing that you measure is in proportion to how large the system is, right? So if you are trying to measure a system like the brain with the technology that you have today, you're not going to be measuring. You're going to be the probability that you have many confounders that you're not measuring just dramatically skyrockets as you move to larger and larger systems. So that's one part. And then when you're trying to discover networks from data, the sample size that you need, the trials that you need to reliably discover these relationships that give you the network structure also that raises dramatically, because you have noise in every one of these things that you measure, and this is amplified by the number of things that you measure.
Anybody else?
Yeah, I would add to that in that it's, you know, one of the basic issues with causal inference in large dense networks is that even before you infer the directionality, a basic thing you need to be able to infer is like the conditional dependence relationships between the individual units of interest. And as a network gets more dense, even inferring those, even the non-causal dependencies are harder to estimate in data. And so you have like a dual-pronged issue in that even the non-causal information is very high dimensional, so you need enough data to infer that. And on top of it, you want to infer the directionality as well, so you just have an even higher dimensional problem. And so I mean, and the confounding just makes that even more complex, right? But
yeah, and I want to give people like a little bit of a calibration here. You know, like a neuron might receive input from maybe a 5,000 other neurons. Now let's see if we could even find out what an individual neuron does. You know, like, so if we would have full access to the system, so causality wasn't an issue. I basically patch clamp that neuron, stimulate the presynaptic neurons. I could like iterate nicely through all these 5,000 potentially presynaptic neurons, and maybe I'll be able to estimate what those weights are. Now, that would be 5,000 experiments that are long enough that I can estimate the weight between two neurons. Anyone who's ever estimated the weight between two neurons, that is like highly non-trivial. It takes a long time to get that right. And that is the first step, no? Like, but that is if everything is trivial, no? Like if the neuron is just linearly adding up things. Now, if we allow neurons to be nonlinear, things are much worse. If we allow neurons to, if we don't allow ourselves to basically define every presynaptic input, but we have to reverse engineer the system based on the rest, it's even harder. Now you can say 5,000 synapses, or like 5,000 presynaptic neurons. Maybe I'd need a minute to identify each of them. Well, that would be 5,000 minutes. It's a long time. Add a massive extra factor if we allow them to be nonlinear. Add another extra massive factor if we need to do observational causality, because like that's one of the things that we understand in causality. We basically get to have causally better estimators, but we need more data to estimate it. So ultimately, we are in a domain where it might be impossible to even learn what a single neuron really does with its inputs.
Can I also say something? Oh, sorry.
Go ahead, Bijan.
Well, I was just going to say, this is a very critical issue. One of the biggest problems we had in executing our study, we had hundreds of electrodes in different places, you know, tens of thousands of pairs of sites. It took ages to do the experiment. And we couldn't do the really interesting ones we wanted to do because we ran out of time. And, you know, there's an animal in this whole thing, watching TV or doing a task. And, you know, they're only doing that for a certain amount of time every day. So then, yeah, we dream of wireless interfaces with around the clock, you know, large scale access, but time doesn't scale super linearly or exponentially. It scales linearly, right? And this is really annoying. And so you have to find a different way. And so I think the solution is to go adaptive. And so this is the one way you could do is all model based, right? You could start constructing, you know, sparse assumptions, allow you to do lots of clever things. And we pulsing multiple places in random patterns. Right, so there are a lot of ways you can take some mathematical assumption and then study a high dimensional system if you assume sparsity. And that may or may not be good. And then, of course, you have all the other biological factors that kick in when you start stimulating places simultaneously, right? That's like not for free. Some of those places will be close to each other. And so I think that while those are important things to do, sort of the offline accelerations with a clever experimental design that make some assumptions that you can at least test and validate, but allow you to do the experiment in a more deep way, like instead of doing like, you know, we did sender receiver modulator at triples, right? Maybe we wanted to have like, I don't know, 50,000 nodes in our networks that we're selectively understanding because there are so many different simultaneous mechanisms allowing us to even do this in the first place, right? And so we have to take seriously the scaling problem. And we're dead, right? So then what we do is to go into the experiment. So you search. So everything now becomes, instead of doing offline batch mode stuff, it becomes a brain-machine interface with an interpretive component, which is actually interpreting the network it's studying. And then it probes the network through some sequence of steps that refine gradually hypotheses about what could be going on. And that, I think, is the only, well, it's the best way to go forward. Because then you're at least controlling for your interpretations as you go along, and you're not sort of resigned to something, right? I think the problem with the model-based stuff is you're kind of resigned to the model. If it doesn't work, then you're kind of like, right? But if it's adaptive, then you hope you can, I'm not resigned to thinking we won't find it. That resignation has not happened, right? So I think you can figure it out. I think you just have to search for it. And so that's where we come at this problem.
So Sofia, you were also going to say something about this.
Yeah, I wanted to say something that's quite different, I think, and I've been thinking about it since I was a postdoc at your lab, and because all the questions are about neuron and causality, I think I'm not quite sure, like let's say that you could measure every neuron, you had all the time in the world, you had infinite resources. I'm still not quite sure. You could find how neurons interact with each other, but I don't know if you would be able to explain the higher level phenomena, right? This is a reductionist approach that I don't know that applies necessarily. So I think this is something that people should also keep in mind. Maybe, explaining what happens in all the neurons doesn't explain what happens in the brain or how people think. This is a different level of causality that you may want to think. And this, I think, is related also to a question that I saw somewhere that was talking about.
Yeah, I think it relates to Alexander Ivanov's question about if you get infinitely amount of data, can we solve this problem or something in a way?
Yeah, no, I was talking about something else that was saying that there is, people reason, they have. That also relates to this, I think. But I think, I've been thinking about it since I was a postdoc in Konrad's lab, and I was thinking, why do we really want, is this what we need? Do we need to map the entire connection of every neuron to every other neuron in the brain?
And maybe we should read Farid's question here. So Farid Abuhab asked this very interesting question. Despite having billions of neurons / synapses as humans, we seem to be able to make causal inferences about human behavior. Now, Bijan doesn't talk with me because I said something mean to him. Okay, I didn't, and we're still talking, and we're let me be clear about it. But we make these kinds of statements many times. And then let me read the rest of the question. Human behavior exists in a low-dimensional space. Can we leverage some lower-dimensional space to model and influence human behavior without worrying about brains? It's a great question.
That was the question that I was.
I
think that's related.
Yeah.
Wow.
I like that answer.
There's levels of analysis and there's levels of description.
Yes.
This is psychology and brain, right? This is fundamental.
Okay, but let's
yes, we can. We would have a model of, so we would have a cognitive machine, right? So the model would be, absolutely we can do this, right? And you do this through, so I would argue you would do this through psychology, where you would have some mental representation of the behavior, right? This is, the observable is always the behavior, right? And at least until you get some, advanced technology that shows you something else. But for everybody, the observable is the behavior. And then the explanation is, not necessarily neural mechanisms, although that could be the explanation, but it could absolutely just as well be at the level of the mental representations or the psychological representations. And they're, they're very explanatory. That's,
right, but then the question is, if we think through the question a level further, at some level, at least what I read behind that question is, well, why are we even focused on the level of individual neurons? Now, you could say we can potentially build meaningful theories at the normative, the Y level, where we are basically, let's assume the brain is really good at solving that. And in that case, we don't need to demand causality. So I think there's a bigger conceptual debate behind the question. Very interesting.
I didn't follow that. Thank you.
Yeah, and I want to sort of kind of add this, that it also kind of depends what the goal is. If, for example, the goal is to make changes to the system at a neural level. If you want to fix something, if you want to fix a broken system, then we have no choice other than breaking the system and understanding what's inside it. So I think like it really depends what the goal is. The goal is just behavior and input output, then sure, but that
okay, but what I'm
the...
saying is that the system may have some emerging properties that even if you break down the system and you find out what each of the parts does, it doesn't mean, it's not necessarily a clock. That's what I mean. It may have emerging properties. Even if you break it down, you don't necessarily understand what are the emerging properties in the next level. I don't know. I mean, this is obviously I have a very shallow, this is it's not a very deep opinion that I have on the matter, but I've just been thinking about it.
So this is really interesting, right? Because everything, when it's causal, gets down to quantum mechanics, okay? Is what, at least for electromechanical. The stuff we're talking about with neurons and chemical stuff, it's not gravitation. And that is not really what we mean when we say a causal mechanism, is it? We don't talk about that anymore. We've already given up on like the level of atoms and so forth. We're already at the level of molecules or cells or other things. And we do obsess about the neuron. I think it's a wonderful obsession. They're really pretty. There's lots of them. But
yeah, you're
why is that the causal level? Why is that the causal level? What happened that Konrad can say, oh, my normative thing's not causal? I don't think you need to say that. I think the point is that it could causally, well, I'm not going to say which specific theory we're going to describe or level of description we're necessarily going to describe, but I do think that there are mechanisms of the mind that are causal to behavior, that you can't stop yourself from doing it.
Yeah, like I might say something because I'm envious about something.
And you can't
like
control
the explanation it. I said that because I was envious of something gets to be feels like a perfectly valid causal model. It's the kind of it's a kind of statement that humans will very frequently make, starting in
the issue is we don't really understand how those things work. We're all armchair causalists, causal reasoners. Causal reasoners. There's a baby, this is well known. The point in that question is well taken, but I don't think that it means that they're not descriptions of the mechanism descriptions we have that lack causal power. We just don't necessarily understand how they work. And I think this is exactly what Sofia, I would think that this is what Sofia is saying to me. I hear anyway. Yeah, we don't need to go all the way down to that level. There's this emergent thing, it's called the mind.
I'm not saying that we don't need to go down to that level. I'm saying even if we go all the way on that level, doesn't mean that we get, that we explain the next level completely.
That's true.
That's what I'm saying. But I have no evidence for that, obviously. But I know in other systems, like in physics and in molecular biology, many times it has proven to be so, right? So
Manjari wants to say something. I want to have her. Yeah.
Yeah. I want to reiterate that I agree with the with everybody who is saying that mental thoughts. I definitely believe that mental thoughts have their own kind of causal power, and I totally agree with Bijan that what we don't understand is why is it that a lot of us interested in mental phenomena know that, or we hypothesize that, of course, mental states likely have a causal impact on the brain itself. But the question is, how does this happen? And I think Peter Shea at Dartmouth has some very interesting things to defend the existence of mental causation and how we might study it. And one of the things he argues is that, well, at some level, mental causation means that, it has an impact at the synaptic level, and therefore we should be interested. Maybe even though you are interested in emergent causation, ultimately that has an impact on lower level units. And that's the kind of thing one would be interested in understanding how it happens. And so, yes, we want to study causes at different levels of the brain, but you also, so you don't want to be reductionist, but at the same time, even studying the lower levels doesn't mean that you're giving up on the higher levels either. So
yeah, completely agree.
Okay, I'm going to move on to another set of questions. I'm just like going through the question and kind of combining them into one question. I hope I capture all of it. I think there's a whole bunch of questions related to correlation on methods, basically. Granger causality and correlation fMRI connectivity and stuff like that. And how they can tell us about causality. Do they tell us anything? Do they not tell us anything? Is the problem that we just don't have enough data? And basically, I kind of like combined pretty much three questions from Alexander Ivanov and I think Emily Avery Jones, can we infer causality from neural recordings? These are all kind of deeply connected questions. So why don't you all answer it? There's one more question I forgot, yeah.
Yeah, right. So I think the problem with inferring causality from observational data is that you can rarely rule out the possibility of confounders. If you have confounders, you're not going to figure out what you're finding is not going to be a causal model necessarily. There are ways you can rule out some causal models, right? That's the idea. When you have the observational data, you can rule out some causal models. It is very rare that your observational data will give you one single causal model that is possible. So if you cannot rule out the possibility of confounders, you cannot infer reliably, or you cannot be sure that you have inferred the correct causal model. And I think the larger the system is and the less intuition you have about it. So I work in medicine, for example. My examples when I apply my algorithms are usually clinical or biological. In some of these cases, you may have some intuitions about, have I measured enough variables? Do these seem to be covering all the confounders? I think in neural data, this must be less the case because you have no idea, I think, in general. So I think it's very unlikely that you can say for sure, I have found the causal model using neural data. That would be my short answer.
But hold on, there's one thing that I wanted to emphasize with respect to Alex's previous question. So it's really interesting what happens if you get more data. If you have a real causal techniques like instrumental variables, if they're justified like any of the real causal techniques, then you will be noisy, but as you get more and more data, you will converge on the right value. So it goes like in most cases, because it's 1/square root of the number of samples is how your error goes down. If you have any of the other techniques like Granger or partial correlations or any of these, it doesn't go to the right value. It goes to some value that is not correct. And that difference between where it converges to and what reality is, that difference may be arbitrarily large.
Okay, anyone else? Okay, I'm gonna move on. This is a question, I think we covered instrumental variables, so I'm not, Therese, I'm not going to answer your questions. I want to move on to the next question, which is actually kind of related to Kas's talk. It's kind of moves away from causality, but I'd like to ask that because I kind of like these questions. How does one thread the line between practicing responsible science, not exaggerating results, conflating terms, and practicing statistical rigor, and producing results and attract funding and etc.? Who wants to get this?
I don't think anyone has the answer to that question.
Yeah.
Okay, but I have strong opinions there. So let me a little bit there. So we are all doing science because we really care about understanding brains. Now, like I hope that every last person who's at Neuromatch Academy does it because they really want to understand brains. And if not, that's kind of sad. And in that situation, we don't really have a choice but try and do the best science that we can. And if there is a wrong set of incentives, which might be there, like a lot of papers wrongly claim causality, and I can give you so many examples of that, and make it into like strong journals. That doesn't mean, A, it doesn't mean that we should do it. And B, I think the right approach is to change the culture, basically call out people. And I know there's this like, we don't have this culture of calling out things, but I think it is good and we should call out the big leaders in the field. If a big leader in the field makes questionable causal claims, then we should call them out about it. And if we do it, we will at least over like a few years shift the way people on study sections think about it. And I have seen discussions where people basically came around and said like, look, no, it's true. I cannot actually believe this causal statement, and therefore, like, I feel differently about this paper or grant or something. So I think we can fix that, and we should all work on fixing it. And if we don't, that's just sad. This like I know we all have careers, and everyone has a career, but like we are also in it because it's awesome to find out how the brain works, and we shouldn't forget that.
Wait a second. Research scientist is not a career. Okay, this is a vocation. You do this because you have to. Okay, the first thing I seek to evaluate in any potential PhD student is, do they want to do the PhD or not? And then I try to figure out if they need to do the PhD or not. Plenty of people will want to do the PhD thinking that it's going to get them to the next step. But they don't need to do it. They can do something else. And you know what? Not everyone who starts their PhD finishes their PhD.
And there's nothing wrong with that.
And I don't want to be there. No, they often get paid great salaries and they move on.
And they invent things that make the world more beautiful. And
it's not good for the program. Programs that have students that don't graduate are neglectful. This is how it's perceived. Students that enroll and go off and earn 4 times what their advisors were earning, because they learned some really powerful tools that are valuable to society, are considered failures to the graduate program machine. And this is important to the graduate program because we seek to support our students. And there are plenty of places that neglect their students and they don't graduate. So I think the main thing to remember about this is that getting research in neuroscience as opposed to a career is not I don't perceive it at the PhD level. I don't perceive it as being a career move. You're not doing it to get famous. You need to do this, otherwise you will be unhappy about something. And this is how you intend to solve the problem, is to get a PhD and explore whatever these ideas are. So I really kind of object to the idea that we need to be becoming famous. You need to have funding. You need to exist. People need to care about what you're doing. But that doesn't mean you need to be famous. And we actually have really great megaphones nowadays that you can use to become potentially famous. And yeah, just put your voice out there. And you can definitely do that. That's not hard. And it's quite effective. So I would be positive about this. I would say, have some ideas and tell us about them.
And I briefly want to react to Alex Ivanov's comment. He said, how can you do that as a junior scientist? Guys, you don't understand your market situation. The market of like good young scientists is relatively small, and lots of great labs will constantly be looking for great PhD students and postdocs. It's you who are powerful. And it's the senior labs who, in a way, need to appeal to your preferences.
Okay, Manjari is very quiet.
I mean, I think the original question was a little bit about how do you deal with
yes.
the hype. I mean, I can just I can't claim to say I know how to deal with hype and also be, you know, a famous scientist or also get funding. I'm still very early in my career in that sense. But, you know, if I couldn't, to the extent that I choose to stay in science, it's because I want to work on intellectual questions that I don't have, that I might not be able to work on anywhere else. And for me, you know, that's like, I can't stay motivated in science if I make pragmatic decisions. I mean, for better or worse, that's the way I am. And I think, you know, I look back historically about, you know, what kinds of science, what kinds of papers like make a difference. I think you have to stay true to the questions you're interested in and saying, are you doing the best job to answer that question? And if not, then what's the point in doing it? So, I mean, that's kind of what drives me. Like if I start making pragmatic considerations of how is this going to sound and let me make this more appealing, then that's the day I leave science. You know, that's how it is. Yeah, that's how I think about it.
I also want to say something as a junior faculty. I think, you know, whichever way you see it, it's okay. It's fine if you see it as a career. It's fine if you see it as a job. It's fine if you cannot sleep at night. I think there are ways, I struggled with it a lot in the beginning, and I think there are ways, you can find ways that you can talk about your results and how important they are without lying. That's the bottom line. I think you still, part of the scientific, like part of being a scientist is hyping your results, not hyping them though, is exaggerating how, or not exaggerating, maybe I'm not finding the right word. Highlighting how important the results are because you believe in them, but you don't have to lie about them to have a career. That's what I think. And if it gets to the point where you have to lie about it, you can choose to do something else.
Okay.
Yeah, I really like what Sofia said, and I want to reiterate that. I think there's a way of talking about why the particular result you have without hyping it up is a stepping stone to the next thing, without saying that you're already at the final goal that you're trying to achieve. And I think there's an art to doing that, and maybe it takes a little bit more effort in writing your paper, like your introduction and discussion, than maybe everybody spends on it, but it is possible. So I think there is, like, if you care about it enough, you can structure it that way, and it's never, failed me. So, and I think lots of people do it. So I think it's just a matter of caring enough to do it that way.
Okay, I want to bring us back to causality. That was wonderful. So the next question is from Alan Zhang. How do you establish causality when your variables of interest influence each other bidirectionally? I think that's a good question. Yeah, I don't know the answer.
From my perspective, I'm not sure exactly what you mean. I'm assuming when you have feedback loops, usually time would be able to resolve this naturally somehow. Yeah, in stationary Bayesian causal networks that I work on, for example, you cannot have feedback loops, but if you unroll them over time, a feedback is something natural. That's what I can say from my my point of view. I don't know if that was the question.
Yeah, I think it's like they're imagining like two nodes and one influences the other one and the other one influences back that one. I think that's the question, if I understand it correctly. I feel like Konrad
to amplify Sofia's point, like if we have two nodes that influence one another, one thing we can do is unroll time, where you can basically say it is not actually that those two nodes influence one another. I have the neural activity at every point of time, and they only have forward interactions.
Adding to that further on, if you expand notions of like, dependence to much more sophisticated, for example, like time frequency causal graphical models, a lot of biological phenomenon happen at different time scales. Like causation one way is on a slower time scale, causation the other way is on a faster time scale. And so if you decouple the problem and look at your data in the right domain, you can actually build graphical models, causal graphical models. All of that infrastructure is possible in like not necessarily regular time, but in like time frequency or some other space like that. So it is possible to break these down. I think these areas are like less developed than time domain, but that's those are all cool areas to work in to solve that. And like as long as things are not happening simultaneously at the level or scale that you're studying things, there's like a way around it.
I agree.
Bijan, do you want to say something? Or?
This is a really complicated issue. I think that levels of analysis are the only way to solve anything like this, because if you've got directly interacting microscopic elements that are constantly affecting each other in a strong manner, good luck. Saying anything causes anything. Right? This is not relevant. And then you're dealing with the emergent level of the dynamics and all of that stuff, which is perfectly fine. And then what you want to do is have causal models of this, which actually describe how the system responds to causal manipulations, perturbations, which is not the same thing as a separate in causality, but becomes the name of the game. Right? So now what you're doing is you're describing the system that has these emergent dynamics that are laws of motion, basically. You need to infer or somehow learn. And then you manipulate the system, and then you say that it changed its performance somehow. You think something differently, or you see something differently. And then this is a causal test. But you're not inferring causality at the level of discussing directionality between neurons because they're all connected so strongly. You can't do that. Now, there are in between levels of this, of course. We have long range connections, or the scaling of synaptic strength along long distances as well. The scaling of effective strength, and the number of connections over long distances has been well studied. It's complicated. We don't really know what it is, but it definitely falls off. And so you do get a degree of coupling that varies over distance. And so then maybe if you do the right manipulations, you can come up with the right ideas. But I think that it's very complicated, and it needs to be divided into different scales and respects those constraints.
Okay, thank you. I'm going to move on to the next question, and I'm going to augment it a little bit. The question is from Jake Gavinus, and he asks, how should we think about causality in a large scale sense? Like when we think about fMRI or EEG data, can we apply the methods we learned today for perturbation studies like TMS, TDCS? And what are the I'm augmenting it by asking, like, what are the things we need to consider when we go to that level? What are the complications that when you look at the network at a larger scale, you have to kind of take into account? Anybody?
Carefully.
I mean, the first question is, can you do it or not? I think that's the simple question first, and then.
Can we apply the methods learned today?
Yeah.
Definitely. I think the most exciting thing about methods is that they can be, in neuroscience, they can be applied across scales so fruitfully. There are important issues you need to keep track of because everything gets complicated in terms of the inferences and the interpretations, and that's where you get kind of called out, especially. This is wrong, for reasons that you probably should have educated yourself about. But that doesn't mean that the method's invalid.
But let me seed a little bit of uncertainty here. So if we want to define something like causal influences between areas, which is ultimately behind the question, then that in a way is already a commitment to the idea that that makes sense. Now, like, let's say visual system. Why would I have more activity, say, in object areas if I have more activity in V1? Well, maybe not. Now, like within an area, it might matter greatly which neurons are active. So at some level, the idea of a connectivity between brain areas that can have like a sign where we can say, if I TMS here, this thing goes up, is something that forces us to make a certain theory statement about it. And like we need to bring a theory that says the average activity in this brain area matters. And that may or may not be true, but like I just want to say, like, we cannot be sure that results from these techniques actually are meaningful.
Well, the meaning comes from what you say about the result. And I said that was subject to interpretational constraints. So absolutely, be careful not to put your foot in your mouth. The problem with EEG is it doesn't give you spatial resolution. So, you're not studying that question with that measure. So yeah, if someone wants to sit around talking about how brain areas are talking to each other with a bunch of, in a regionally resolved manner, you can do it based on the temporal properties in some important ways. But yeah, you're not going to say this group of neurons is talking to that group of neurons based on EEG measurement. Maybe if you've focused on the neurons involved in the auditory evoked potential, or you're focusing on the neurons in one hemisphere versus another, and even that's a little complicated. So yeah, don't do that. But fMRI is totally spatially resolved. And now you have another set of very important interpretation limitations, which is slow and it's not measuring neural activity directly. And we all hear this all the time. That doesn't mean that fMRI connectivity graphs are meaningless. And I think one of the most exciting things is the work, one of the most, a very exciting set of observations, influential in my thinking in this area, is David Van Essen's published work, whose first off the name I cannot remember. I apologize, but the work, I recall the following facets of the results, which was when they looked at the resting state correlations in the bold signals they were measuring. They got these networks that were reflected in the anatomy and they had reliability and all these things. And they didn't need the task responses. And I thought that was really interesting. It was the resting state dynamics which contained the network structures. And the task responses themselves were additional. They were useful. It was not like they were not important. but they really got a lot of information out of the resting state now. And I think of that as being reflective of causal influences that are really sort of strong, that are being measured by those measurements. How they're measuring them and why, I mean, I have no idea necessarily. But I think the method can be applied. It's the interpretation that's the problem.
Sophia, Manjari. Yeah.
Yeah, I know, I totally agree. I work a lot, you know, all the methods you learned today definitely applicable in the large scale, you know, with the caveat that if you're working with regular functional MRI and you're not doing anything beyond that, then you have to say that you're talking about bold activity in one area causing bold activity in another area. You can't necessarily make, you know, assume that that is neural or neuronal firing, right? You have to start getting into all these other types of techniques, you know, multimodal techniques, techniques that are measuring pure cerebral vascular activity and so forth, if you really want to uncouple, you know, what explains the bold to bold associations. But I think you can change your question to be causally meaningful in the appropriate way. So that's certainly how I view, you know, looking at causal influences in fMRI. You know, I restrict my thinking to, you know, can we really say area, bold activity in area A is, you know, has the capacity to causally manipulate area B, or is happening at a particular, in a particular experiment. And I think all the tools that you've learned today are very useful in trying to eliminate source of confounding and trying to answer that question very rigorously. Yeah, let me stop there.
But let me take the unpopular opinion here. So functional connectivity is very attractive. Like, it's a set of techniques. In goes, say, an MRI signal, and out goes the network. I would argue that we at the moment have no evidence that the networks are really meaningful or even better than chance. We just haven't seen studies that really clarified that. So I know that we want causality, and like, we can agree, like, I really want to understand causal interactions, but it is not sure that it is possible with fMRI. And we need to be aware of that.
I will agree with that statement. I will agree with that. I think the networks are real, but I don't think that they're resolving. But I don't think that, but I've never measured one and published one myself, so I can't tell you statistically significant. Yeah, I mean, how bold causes bold is pretty complicated. I would avoid getting too carried away there. It's an inference about a mechanism which could affect, you know, I think that the way Manjari talked about it is right.
To complete answering the original question about what are the problems you should be aware of, I think that when you're studying neurons, the neuron is a well-defined unit. At the large scale, what you're calling regions is like highly tentative. And once you start wanting to take causality seriously, if your node does not represent a homogeneous unit in reality, then your causal inferences about, you know, A causing B when A is not what causal inference statisticians will call, you know, is it like a consistent and monotonic variable? Like, they have certain requirements for answering causation. And if it turns out that you're actually calling, you know, very heterogeneous mix of things a single unit, then the notion of that being a causal variable is itself suspect. And so I think that's, and in fact, probably a very fruitful area of future research. Everybody's interested in testing these assumptions of what are these brain areas and regions that we are trying to draw causal inferences between. And I think it's a very, so far neglected, but very important issue in the large scale situation.
Okay, I'm gonna ask the next question. Yeah, before asking the next question, it's 8 already. So I think we can stay a little bit longer and ask a couple more questions, but if anyone wants to leave, please feel free to leave. You don't have to. Oh, what? Sorry.
I have to leave soon.
Oh, I'm so sorry. Okay, Konrad, I'm passing this decision to you. What should we do?
I don't know. I think we should then break off here. Not like if
Well, we should have another question. I don't want to cut it off prematurely, but I have a. I'm willing to be 10 minutes late to everything.
Cool.
Okay.
So why don't we have another 1 or 2 questions, or maybe 1 question, and then we could give.
Okay, I'm going to ask one question and also augment it with my own little bit of flair. So the question is from Vaishnavi Sukumar. Sorry if I'm butchering your name. Why do we so rarely see instrumental variables used if they're so much better than regression methods for partially observed data? And I'm going to actually sort of augment that, is that why do we have so little causal data in causal experiment in general compared to the large body of correlational studies?
I want to answer that question.
Go ahead.
They're hard.
That's right.
The first thing that you have to remember when you're doing a causal experiment is you're manipulating the thing while you watch it. That's hard.
Yes.
We're figuring out how to do it in different ways. And we have more and more powerful ways of doing it. And we're, it's great. But let's not forget, and let's remember that it's hard to combine things. I talk to people making microscopes. They want to, make beautiful pictures of volumes at high rate with great resolution and all kinds of cool stuff. I also want to stimulate while I record. Sorry, dude.
For instrumental variables, if I may say, it's very hard to find a good instrument. It's not given that for something, for your outcome of preference, that there's going to be a good instrument. That's pretty rare.
So I want to say instrumental variables are actually very, very frequently used, just not in neuroscience. It turns out that economics about 30, 40 years ago looked at what they were doing and basically realized that they're making causal statements, which is like, should tax rates go up or down, and policies, and also and so on and so forth. And it wasn't getting them anywhere. They wrote a paper that's called, let's take the con out of econometrics. And what they've done since is they said, we only want to, if we have causal questions, we only want to accept causal answers to that. And they cleaned up their act. And now if you want to publish in a good econ journal, you're expected to use a causal method. You're expected to use instrumental variables or difference and difference, or any of the other quasi-experimental techniques. And otherwise, you have to be very careful about arguing that you may have causality. I think it's simply time that neuroscience follows the same path.
Manjari, do you want to say something?
I don't know, Konrad. I mean, so I'm not sure, because, for example, if you look at clinical medicine, people there think that unless you do an RCT, you can't draw any causal conclusion. But it turns out that if you have a properly designed observational study that carefully thinks about the causal question of interest and designs a study such that it has the capacity to mimic the RCT, then it can answer that question. But I think it's hard to set a science. I hesitate to support setting some kind of science-wide standard, evidential standard that then, kind of keeps out things that are in fact useful. I mean, that tends to happen in a field. So I don't know.
Okay,
I'm totally sympathetic to what you're saying, but at the same time,
I apologize if I overstated the case. All I want is us as neuroscientists to start taking causality really seriously, which means if we run a correlational study, we should give very good reasons why we think this is a good approximation to causality. And we should discuss that in all of our papers. And when we can, we should use causal methods.
I also think it's not just that there's not as many causal studies. The other problem is that most causal studies are basically confirmatory. So you basically run a whole bunch of correlational studies, and then you basically know what the outcome of the causal study is. If it didn't turn out that way, you basically are not publishing it. So it's not. I think there's some other factor here that also needs to change, and I don't exactly know how that should happen. Yeah. Yeah. Okay.
If there is time, I would like
Sofia?
to answer the question about the socioeconomic status and tobacco taxes. because I think it's pretty specific.
Yes, I think any questions, detailed questions about the material in the tutorials, we could actually take it to the Neurostars. Please post your questions on Neurostar, okay. And then yeah, we can all go there and answer questions on Neurostars. And I think there's basically no time to answer any more questions, so please, all of your questions, post them on Neurostars. There'll be someone to engage and answer your questions. And I would like to thank Manjari, Sofia, and Bijan for this, and Konrad, obviously, for this really fun discussion. I had fun.
And we should also thank Maryam for just fantastic hosting here for us. Thanks so much.
Thank you.
Oh.
Bye, everybody. Bye-bye.
Bye.
Everybody, it was a lot of fun.
Yeah, bye-bye.